Hello everyone and welcome to today's webinar, See When Your AAV Can't Stand the Heat with Uncle. I am Kaylee Bach of LabRoots and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Unchained Labs. To learn more, please visit unchainedlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Dr. Kevin Lance, Director of Analytics Marketing at Unchained Labs. Kevin, you may now begin your presentation.
Welcome to my talk, See When Your AAV Can't Stand the Heat with Uncle. Unchained Labs has been helping out with our gene therapy squad for all kinds of problems and all kinds of gene therapy tech. We've been honing our tools to deliver answers and save boatloads of time for AAV and other viruses. Stunner quickly delivers answers on titer and empty flow ratio. Big Tuna takes care of buffer exchange and concentration. And Uncle is the key to all kinds of stability insights. That's exactly what we'll be checking out today for AAV. This is one way to think about AAV. If we think of AAV as a piñata, the DNA is candy and the capsid is cardboard and colored paper, then everyone wants a lot of information about that AAV. And current analytical methods aren't good enough. But Uncle helps you know exactly how stable your AAV is and how to help stop aggregation from becoming a problem. So if we keep thinking of AAV as a piñata, then this is what a thermal stability experiment looks like. Uncle is that broadsword-wielding warrior who is wasting no time seeing what this capsid can withstand. Meanwhile, gene therapy researchers are there in the background, cheering on the experiment and excited for the results. So let's learn a little bit about Uncle. Uncle is an all-in-one stability platform ready for AAV. It uses two lasers to excite either label-free capsid protein intrinsic fluorescence or a variety of dye-based fluorescence. We'll actually use both fluorescence methods to look at AAV stability. With full spectrum fluorescence detection, Uncle can pick up on both kinds of those signals, which is really valuable. SLS is the scattered light from those two lasers and gives information on when aggregates form, especially during a thermal stability ramp. DLS is dynamic light scattering and allows for detailed measurements of size and size distribution. Those three detection methods can be operated isothermally or across a heat ramp designed to thermally stress samples and see which ones can take the heat. That means Uncle's able to deliver results on aggregation and two different looks on viral capsid stability in about 2 hours with only 9 microliters of sample for up to 48 samples at a time. The secret sauce of the Uncle is the uni. It's an anodized aluminum frame with 16 quartz cuvettes, where each quartz cuvette holds 9 microliters of sample. So all you do is pipette in your sample and seal the uni inside of its blue frame so silicon seals clip around those little cuvettes. Just like that, you have a nicely sealed sample where you don't have to worry about evaporation over long periods of time or over a thermal ramp. This is also a handy sample holder because you can pop it into, say, a 37-degree incubator and do a weeks or months long stability experiment right in the uni, or you could put it into a freezer and go through freeze-thaw cycles also right in that uni. Those are just some advantages to having a well-sealed sample holder with such a small sample volume needed. Okay, so coming back to AAV, Uncle can deliver two different kinds of stability information when it comes to AAV: capsid stability and aggregation. When it comes to capsid stability, Uncle will use its fluorescence to measure two different behaviors. On top, you see a cartoon of protein unfolding and capsid disruption occurring as a result, or on the bottom of the left side of the slide, genome ejection can actually occur from mostly intact capsids, sometimes called uncoating, but since this is happening outside of cells, I prefer to call it genome ejection. So we'll look at data both from the literature and from Uncle to demonstrate those behaviors and investigate them further. On the right side of the slide, we'll talk about aggregation, which with Uncle's SLS and DLS capabilities, we'll be able to identify when aggregation occurs, which can be at either at time 0 before an experiment, where you want to check out that your sample is monodisperse, and to know if you should go forward with your experiment. Or with SLS, you can see how aggregation occurs during a thermal ramp as a measure of the colloidal stability of your AAV. So the first question to address usually is, how do we know that AAV capsids have these two different stability options for sort of falling apart, the capsid disruption and the genome ejection pathway? Well, for that we go to the literature. Here we can see 3 different morphologies of AAV in an AFM data set done on AAV8 and AAV9. These AFM images show both behaviors directly. In this case, the AFM data were captured after heating AAV to various temperatures. So at the top, we can see intact capsids as bright circles. And going down the slide to the green boxes, we can see those bright white circles with ejected DNA coming out of each one of them. The authors of this paper also point out that the length of DNA exiting the capsids is variable, and so that supports the idea that this DNA is in the process of exiting the capsid. On the bottom is compact DNA clusters without those AAV bright white circles or capsids. So this is what would be left behind after complete AAV disruption. So in this one data set, you can see sort of all 3 states of AAV, both the intact capsid form, the form where it's sort of pseudo-stable and ejecting its linearized DNA, and then the finally totally disrupted pile of DNA form. All right, so to check out capsid stability, Uncle will make use of the intrinsic fluorescence of the AAV capsid proteins. Uncle's going to excite those capsid proteins with its 266 nanometer laser, and the tryptophan and tyrosine residues of those proteins will then fluoresce. When those proteins unfold, that fluorescence changes in a few characteristic ways and lets us know unfolding has occurred. Here we have a quick look at the intrinsic fluorescence signal collected by Uncle on a sample of 1 × 10⁻³ AAV9 without SYBR Gold. Each line here is a fluorescence read at a different temperature, starting from the top and going to the bottom. As the sample temperature increases, the signal intensity drops and shifts to the right, which is called a redshift. So that blue arrow and dashed line are meant to guide the eye to this redshift by showing you where the true vertical is and then showing you where the peak of each of those distributions is. Also note that you can see the SLS signal from both the 266 nanometer and the 473 nanometer lasers on this fluorescence trace. So if we take that same intrinsic fluorescence data, we analyze it to look for the sort of spectral center of mass, also called the barycentric mean wavelength or BCM wavelength, we have the data shown in blue. So as you heat up the AAV sample, at a certain point, those proteins start to show that red shift, and that BCM wavelength increases. And we can identify the onset temperature for that increase, here at 74.2 degrees Celsius, and the melting temperature for that unfolding behavior, here at 78.6 degrees. At the same time, by tracking the SLS signal from the 266 nanometer laser, we can see aggregation behavior, because when AAV is just floating around happily, that SLS signal will be quite stable, but once aggregation has begun, that SLS signal increases. So you can see in green that SLS at 266 signal increases with the T-ag or aggregation temperature at 74.5 degrees. So with this one run, we're able to look at both protein capsid unfolding and aggregation through the means of looking at simultaneous fluorescence and SLS. For genome ejection, Uncle will make use of SYBR Gold dye, and will excite SYBR Gold with its 473 nanometer laser. And the purpose of adding that dye is to detect genome ejection by showing an increase in fluorescence signal. So at the start, SYBR Gold will have a low fluorescence while the genome is inside of the capsid, but any free DNA will show fluorescence. But as the capsid is heated and releases DNA, then that fluorescence of SYBR Gold will increase because the SYBR Gold dye and the DNA can now interact with each other. The temperature where the dye fluorescence increases will be the stability readout. So here we have a sample of 1 × 10⁻13 AAV9 with SYBR Gold, where we can see the dye's fluorescence in a different part of the spectrum from the intrinsic protein fluorescence. So that's between about 500 and 650 nanometers, where that fluorescence signal will increase as temperature goes up. We also see the SLS peak at 473 nanometers. In this experiment, only that 473 nanometer blue laser is used, so that's why there's no intrinsic fluorescence signal or SLS signal at 266 nanometers. Here's a look at the analysis for that SYBR Gold fluorescence. Again, we're looking at fluorescence in green, but this time it's the area under the curve. So the headline of this experiment will be that large increase that you see. But let's walk through it from left to right. So starting at room temperature at 25 degrees Celsius, what we first notice is that there's some signal from existing free-floating DNA present with SYBR Gold dye in our AAV sample. So that's the kind of thing that we can purify out with a benzonase step followed by a molecular wave cutoff, buffer exchange, or dialysis, or running on Big Tuna. As temperature increases, what we see is first that SYBR Gold fluorescence decreases because there's a relationship between temperature and SYBR Gold fluorescence. But starting at about 50 degrees Celsius, we see a large increase in fluorescence and an identified melting temperature at about 58 degrees Celsius. So this is that headline of the experiment showing the ejection of viral DNA at those temperatures, consistent with literature data that we'll see a little bit forward in the experiment, and also with that AFM data that we showed earlier. Simultaneous with this SYBR Gold fluorescence ejection, we're also looking at the SLS data in blue, and this is showing our aggregation onset, again, really close to 75 degrees Celsius. So with one experiment, we're seeing the amount of DNA that's present at the start of the experiment before any AAV have started to degrade because of temperature. We're seeing the temperature at which DNA starts to release from our capsids, and we're seeing when aggregation begins via SLS. As we read tons of samples on the uncle, we've noticed that each serotype behaves very differently. So here I've shown a series where we've mixed together empty and full capsids for AAV5 on the right and AAV9 on the left. What you can see is a couple of different observations. First, AAV9 shows sort of a bimodal genome ejection behavior, but that first peak between about 40 and 50 degrees Celsius is rather weak, and then most genome ejection appears to occur between 50 and 60 degrees Celsius. Contrast that with AAV5, and you see a much more clear behavior where you get a lot of genome ejection occurring between 40 and 50 degrees Celsius, and a little bit less occurring at higher temperatures. You also notice that for both of these samples, as we increase the number of empty capsids or decrease the percent full present in the sample, that the signal coming from SYBR Gold also decreases, which is what you expect because, hey, there's less DNA present in the sample. If you check out a couple more interesting serotypes, you can see the extremes of these aggregation and genome ejection behaviors, where on the left we're looking at php.eb, and at the right we're looking at ancient 80. And you'll notice these are pretty unique in that they're at extreme ends of the spectrum for the serotypes that we've tested. So with php.eb, the aggregation temperature lines up exactly with the genome ejection temperature. But with ancient 80, it's actually the largest difference between those two behaviors that we've seen, where there's a 30 degrees Celsius gap between the genome ejection and aggregation behaviors. And importantly, that something that we saw earlier is that aggregation behavior always syncs up with the protein unfolding behavior from the capsid. So even though we're not looking directly at the protein behavior with this blue laser and SYBR Gold dye, we can infer where protein unfolding occurs because that will also line up with the onset of aggregation. If we want to check out going beyond amino acid sequence and serotypes, we can look at excipients and formulation, where here we're looking at the intrinsic fluorescence and protein unfolding on the left, and the SYBR Gold fluorescence and genome ejection on the right. In green, we have a PBS sample, and in blue, we've spiked in arginine into the buffer of that sample. So here, that high concentration arginine spike decreases the protein stability fluorescence by about 7 degrees, but actually decreases the genome ejection stability by about 13 degrees Celsius. So that tells us a couple of things. First of all, that formulations do impact both stability behaviors, and that they impact those stability behaviors to varying extents. So it's important to measure both stability behaviors, which you can do very nicely and easily on the uncle. Because you can't just look at one and see that it's a 7 degree C difference and extrapolate that to the other behavior. It's something that you do need to get both data points independently. Okay, so now we know we can crack open the AAV pinata. Let's also take a look at what you can do to change how things come apart with a further look at the impact of formulations. So we'll take a quick pit stop in the literature to get something to compare this with. So in this paper by Bennett, a variety of buffers were tested, and it's apparent that the choice of buffer has an impact on protein unfolding and AAV stability. So here I'm highlighting AAV serotypes 2, 8, and 9 because we'll check those out on uncle a little bit later. In this case, the authors are using a DSF method using cypher orange dye that does look specifically at the protein unfolding behavior of AAV capsids. So we'll be comparing our protein intrinsic stability to those different data points shown by Bennett. Okay, so to set this experiment up, what we did was use Big Tuna to buffer exchange those 3 serotypes, 2, 8, and 9, into a variety of buffers that spanned a variety of pHs. And we're doing this in a 96-well format plate with a 10 kilodalton molecular weight cutoff filter. So after that buffer exchange, we then read those samples on uncle to screen for their impact on capsid stability and genome ejection. So first, let's start with capsid stability and protein unfolding. On the left, we have AAV2, and on the right, we have AAV9. So what we're seeing here is we're checking out exactly when proteins start to unfold in response to increased thermal stress. So with AAV2, we're seeing, starting with PBS at pH 7.4, the earliest unfolding of those capsid proteins, and then as we get more acidic in our pH, you start to see later and later protein unfolding. And you notice that covers quite a large spread of AAV behavior across temperatures. Meanwhile, on the right side of the slide in AAV9, at those same 4 pHs, we see unfolding of those proteins at very high temperature, and they're also very closely bunched together. So it's very different behavior based on AAV serotype. If we plot for our 3 serotypes, the data where we're looking at different buffers and trying to understand how they behave, what we can see is this relationship between buffer pH and melting temperature of the capsid proteins. And just to the left of each graph, what we can do is, again, add in that data from Bennett and see that pH is a really nice tool that can take AAV stability across the same range of stabilities that Bennett saw in their buffer screen. So this can be one way to start to understand how your AAV might behave once it gets inside of cells and it's inside of a late endosome. Do those proteins start to change their stability behavior? And likewise, you can use it to optimize the buffer that they're being stored in to make sure that they're going to hold on to their capsid protein stability as long as possible. Okay, let's shift gears and look at the other half of the equation for genome ejection. And here we're just looking at AAV2 and seeing the impact of different acetate buffers at 3 different pHs. And you can see at actually more acidic pHs that now we have genome ejection behavior that is sort of more stable, so that's occurring at higher temperatures in those more acidic pHs. And if we kind of wrap everything up in one nice graph, what we can do is check out our AAV2 sample and look at various different pHs and see that pH has very interesting effects on both capsid stability and genome ejection. So by running these samples on UNCLE in just a couple of hours, we can get a very complete look at AAV stability and understand how changes in pH impact both our protein unfolding through intrinsic fluorescence and a look at the capsid stability, and how it impacts our genome ejection behavior of AAV and understanding when our viral vector might release its DNA into the surrounding environment, which could be useful both in terms of buffer storage and in terms of how it behaves in the late endosome of the cells. And also importantly, I want to emphasize that this whole experiment took about 24 hours a day of experimental time. So that buffer exchange on Big Tuna had just a couple of hours, and then 2 rounds on UNCLE with one of them overnight, and we were done with this whole experiment and data set pretty fast. So from all that UNCLE data in the academic literature, we can see that these 2 different stability behaviors happen at different temperatures, with genome ejection actually occurring at temperatures far below capsid disruption. And so this is why measuring viral stability on UNCLE is so important. It can look at both of these pieces of data and aggregation too, and understand exactly what's going on with your AAV sample. So with this data, you can hopefully create an AAV that's ready for battle and won't aggregate or fall apart when under stress. Thanks for your kind attention, and now I'll pause and take questions. But if anyone has any other questions they want to reach out to us, you can find us at info@untitledlabs.com.
Thank you, Kevin, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, let's get started. Our first question asks, do different serotypes have different aggregation behavior?
Yes, so and I would also probably add definitely. So if you're looking at aggregation, there's probably 2 different ways that you can look at that. One is if you just have a sample, bench top, room temperature, and see how quickly that aggregates, and it's something you can very easily check out with DLS. I know, AAVs 1 and 2 are particularly notorious for aggregating quite severely just at room temperature, and AAV 9 is another common serotype that actually is quite stable and tends to not aggregate, and you can get very sharp monodispersed peaks with AAV 9. The other way you can look at aggregation is during a thermal ramp by checking out when SLS increases, like we saw in a lot of the data in the presentation today. And that's going to really stress test the capsid protein stability and going to show exactly when those different capsid proteins unfold and aggregate to kind of get a picture of what might happen to the protein as it starts to, have stability problems in even longer term storage. And that will also differ greatly across serotypes as well.
Great, thank you. This next question asks, how would testing thermal stability help me with my process development?
Okay, yeah, because I think today we spent a lot of time looking at formulation work and talked a little bit about how this can apply to capsid engineering as well. When you're looking at process development, I guess what's really important here is I'm thinking of chromatography steps where you have different buffers going through your AAV, you're exposed to different pHs and ionic strengths, and even temperatures as well if you're using heated columns. So what we saw is across a lot of different buffers and a lot of different pHs, you have varying stabilities. So that might be something when you're considering when you're optimizing your chromatography buffer, especially if you're doing anion exchange chromatography, because using the wrong buffer there can really just expose your AAV to a condition where it's not going to be very stable for very long, especially in the shear stress of a chromatography column. The other thing is when you're using a column heater, some of the AAVs that we've been testing in certain buffers can start to show genome ejection as low as 37 degrees Celsius. So if you're heating your column even up to 37 or 40 degrees Celsius, you could be seeing problems with yield just because of what you're doing with your buffer and your AAV. So answering these kinds of questions about stability for your AAV really helps you understand what your design space should be when making those types of choices for your process.
Great, thank you. All right, this next question asks, can you use Uncle's DLS when looking at AAV stability?
So Uncle's DLS is typically used before and after a thermal ramp for the experiments that I've shown today. And that's just kind of run automatically as a way of checking what the quality of your sample is before running a thermal ramp, and to confirm aggregation through an orthogonal light scattering metric at the end of your thermal ramp. But you can also just use it as a quick DLS check for a low volume DLS system. If you just want to see, hey, I'm going to run some other, cell-based assay on my AAV, and I want to confirm that my AAV is still nicely monodispersed and not suffering from aggregation. So it's a nice little use of DLS. Uncle also is really commonly used for isothermal studies too. So if you want to check out, say at 37 degrees C, 40, 45, 50, something like that, and look at DLS over several days, this is another good way of checking out stability and aggregation on the Uncle.
All right, and our next question asks, can I do this kind of test if iodixonol is present?
So this is an interesting one. It was a result that we didn't really expect when we started testing out these samples. The short answer is yes. If you're using SYBR Gold and you're looking at genome ejection, then you're fine. You're using a blue laser and it excites just that SYBR Gold result, and you see capsid genome ejection as a result, and capsid unfolding can be inferred from the SLS signal. But if you're using UV excitation, iodixonol, you tend to not see any signal at low temperatures, things like room temperature. But then once you start to heat up your sample, you begin to see major iodixonol fluorescence that interferes with that typical tryptophan and tyrosine fluorescence. So if you're looking at signal just at 300, 330, or 350, iodixonol will be fluorescing there if you're using a UV laser to excite it. But it's something you're going to actually miss at room temperature, and it will mess up your results at higher temperatures. So yeah, it's a very interesting situation when you start using iodixonol.
Great, and we're getting great questions from our audience in here. This next one asks, what are the mechanisms for genome injection before capsid disruption?
So this one's interesting because if you dig into the literature and go try to check out all the biophysical studies, it's not been a totally solved mechanism yet. What you can see is if you look at different studies and kind of check out different mutation studies, it seems to imply that there's genome ejection occurring through one of the axes. I believe it is the threefold axis, but I'd have to double check on that. And it's not something that's really been figured out quite yet because it's, the resolution of AFM and other types of microscopy aren't going to be able to give you an answer there. So really you just have to look at kind of mutation studies and seeing where you mutate different parts of the capsid, what is the impact on genome ejection. And that's not really been done in a rigorous way quite yet. But if you sort of start to connect some of the lines, that's where I would look at first if I were trying to study the mechanisms for genome ejection. Is that part of the capsid and the impact of capsid, post-translational modifications and amino acid sequences in that region of the capsid. And that also actually goes along with length and structure of the genome as well.
All right, next question. Can you see glass transition in the thermal ramp?
So typically what we're talking about when we're talking about protein samples and AAV samples is we'll use the words melting temperature, or TM, and that's going to correspond to the protein unfolding temperature. So it's the inflection point between a folded and unfolded protein. And then on the genome side of things, that's going to be the genome ejection temperature. So if glass transition is referring to something else specific, I'm not particularly aware of that term, but yeah, definitely for UV-based protein unfolding, you can see those melting temperatures, and then for SYBR Gold fluorescence, you can see that DNA behavior as well.
All right. Next question, what is the AAV concentration required for measurement in UNCLE?
Okay, so with SYBR Gold fluorescence, looking at those genome ejection assays, the lower limit is about 5 times 10 to the 11th VG per mil. It's the kind of thing you can go a little bit lower. It's just the sort of thing where each genome is slightly different. So if you have a larger genome, you're going to have a lower limit of detection. For a smaller genome, it'll be a little bit higher. For intrinsic fluorescence, it's about 5 times 10 to the 12th. It depends a little bit on sample purity. And then for DLS, it'll be about 5 times 10 to the 11th again.
All right, and it looks like we have time for one more question here. And this last question asks, does this work with self-complementary AAV?
Yes, definitely. And that's actually one of the really interesting parameters that you can test out and see how it impacts capsid stability. So I know generally you're going to pick your genome based on its functional performance, but having a self-complementary AAV versus a single-stranded AAV genome will impact stability, will change genome ejection temperature.
Great. Well, thank you again, Kevin, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Unchained Labs, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.